The broadcast is now starting. All attendees are in listen-only mode.
Good afternoon, everyone. Thank you for joining us. At this time, I'd like to take a minute to review a few housekeeping items to make the presentation run smoothly for all participants. The presentation will last approximately 30 minutes, and following the presentation, we will have a short question and answer session. If at any time during the presentation you have a question, please use the question chat box to type in and submit your questions. We may not have time to answer all of the questions, but any remaining questions will be answered offline. You will receive a link to a recorded version of this webinar in your email within the next week. Again, thank you for attending our webinar today. Without further delay, I present to you Julianne Wolf, the manager of the Biotechnology and Pharmaceutical Division at R.J. Lee Group.
Hello, everyone. I want to take a moment to echo Kathy's gratitude and thank all of you for joining us today. Let me take a minute to just introduce you to R.J. Lee Group. We are an analytical consulting firm started by the visionary scientist you see here, Dr. Richard J. Lee. Over 30 years ago, he saw a need in the analytical community for instrumentation that was more efficient and would provide people with better results or answers. So he formed the company and successfully developed computer-controlled scanning electron microscopy, an automated electron microscopy technique that was highly innovative. In parallel and through the use of this innovative technology to resolve industry problems, the company developed a unique approach to identifying root cause and resolving issues. We call this approach industrial forensics, which goes above and beyond just characterizing the issue, but enables root cause to be assigned and processes to be better understood. We do this by maintaining diversity in our technical staff and in supporting laboratory instrumentation that is equally as diverse. So what we've done in the life science community is to apply industrial forensics to one of the industry's recent and significant issues, which is foreign particulate matter. As evidenced by an increase in warning letters and consent decrees by the US FDA, as well as product recalls, the particulate issue must be addressed in a proactive fashion. You can see some examples of these on the screen here that we've listed for you. It's not by any means a comprehensive report, but just a few examples. So in short, we really feel that we can do much better to build quality back into our products. So now I would just like to introduce today's speaker, Ms. Karen Smith, who is a forensic scientist in every sense. She has brought her educational training together with demands of the life science industry and has become an expert in preparation and characterization of particulate samples, the diversity of particulate investigations, and the application of industrial forensics to these investigations. She participates daily in assisting manufacturers to resolve issues through the application of industrial forensics on various levels and also at different stages of the product life cycle. So without further ado, I present to you Ms. Karen Smith.
Good afternoon, everyone. I'd like to start out by giving a brief overview of today's topics. So first, I'll discuss what we currently use for particulate contamination. Then we'll talk about forensic characterization and how it's utilized for identification of foreign particulate. And then I'll introduce the industrial forensics concept. Finally, I will share 3 case studies to show applications of these concepts in particulate investigations. So first we start by discussing some of the current quality by inspection methods. We have most commonly visual inspection, then there is light obscuration and scattering, and also microscopy techniques. These provide information about the number of particles, the size of particles, and the microscopy techniques can indicate the morphology or shape of the particles. The industry has been using these for years. They are in the USP, they're validated methods. So what is the issue with these current methods? So why do we have to be thinking more about foreign particulate? Here you see 4 particles. Can you speculate what these particles may be or maybe where they have come from? Thinking about the particles with our current methods, they're relatively the same size, they have generally the same irregular shape, and they are all clear, colorless. But we actually have 4 different material types. If you would characterize them, you could get anything from polypropylene to silicon that you see, which means you're dealing with 4 different source samples. you can see the limitation that is present with some of the current reactive methods. So we really need a way of identifying these materials so that the root cause can be determined. So we've established that we need a technique for identifying these materials. What do we require of the analysis method? In many cases, we're dealing with visible, barely visible to subvisible particles. So first and foremost, we're going to need something that's economical in its need for material. We're not going to be able to use traditional bulk techniques. And secondly, we need something that is efficient. We can't wait weeks to get an answer, especially if you have a situation where a product lot is on hold for release. And also, we need a technique that is diverse. There are so many types of materials contained in a manufacturing process, a pharmaceutical process. So can we think of any already established methods in industry that may meet these criteria? What comes to mind is trace evidence analysis from the criminal forensics field. This is an area where small sample sizes are the norm. It's feasible to be talking about small particles of soil, a small paint smear, or a single fiber transfer. The regimen uses established laboratory methods that require very minimal sample preparation. This method is also diverse in touching many different material types, such as fibers, glass, paints, and polymers. So now we have a regimen, an approach to apply for foreign particulate characterization. So let's talk more about the regimen itself. The forensic characterization regimen is analytically a multi-tiered approach, and it uses three major components: optical microscopy, scanning electron microscopy, and Fourier transform infrared spectroscopy. From the optical microscopy, you can learn of the physical properties of a sample. So things like the size, morphology, or shape, and the color. From SEM-EDS, so scanning electron microscopy paired with energy dispersive X-ray spectroscopy, this is a technique utilized to provide the elemental composition of a material. And this is a very well established scientific technique. The third tier of evaluation is determining the molecular composition with FTIR, another spectroscopy technique. FTIR provides information about the sample's functional groups. So once a spectrum is obtained, there are comprehensive spectral libraries that can be utilized in order to make an identification. There are a few additional techniques that can be utilized if additional information happens to be required. So Raman microscopy or Raman spectroscopy, for example, can be utilized when approaching a corrosive type situation or a charred material, and polarized light microscopy can aid to identify glass or various fiber types. And you'll see examples of these utilized later in the case studies. We firmly believe that this type of comprehensive source material characterization in both physical and chemical compositions will be necessary for understanding the potential for foreign particulate contamination in a quality by design environment. So now we have a technique or an approach or a regimen, however you would like to define it. In order to really understand the potential for foreign particulate contamination, you need a holistic approach. So we need to look at a process and be proactive about it. This sounds familiar. It sounds like quality by design. We need to use the principles of quality by design as a model in this instance. We need to be thinking about the particulate immediately, almost concurrent with design. So you can be thinking about potential sources in the process. But the problem with this is looking at the entire process as a whole becomes too overwhelming. So what we need is really a way to compartmentalize, something logical that makes thinking about the entire process more manageable. And this is where the concept of industrial forensics comes in. Its foundation is a holistic approach for systematically looking at a process and extracting information from it. And this falls in line with the quality by design principles. So let's think about a simple example of a process. There are logical sections or compartments to the process. So in most cases, you start with receiving, and then you have the production steps. Maybe there's an intermediate step as listed here, or just a product production step. Then you move on to filling and finishing, and finally packaging. Now, all along the way, you have the potential for contamination from environmental contaminants and secondary sources, which I'll talk about in a minute. So this process example is a pharmaceutical product example, but the same concept can be applied to many different processes, such as medical devices. So we want to use the fundamental idea of industrial forensics to break down the process to think about the potential sources of contamination. So how and why does this make source material identification easier? As we break down the process into components, we will be creating a database. So this database that we will be building can include all or some of the process materials. Some prefer to just build a database of materials that come into contact with the product, where others prefer to build a database with all of the materials in the manufacturing line. The database is unique to the process, and so that makes source sampling become easier. For example, let's look at this process again and think about a list of a few potential materials for each step. So first start with receiving. So you have raw materials coming in. There may be associated drums, buckets, or bags. Then the manufacturing, which probably contains several steps, perhaps involving filtration media, milling and mixing components such as gasket materials or stir bars. And then next you'll have to package the product. So maybe there are plastics involved or foils and inks. These are all going to be the primary potential sources from the process itself. I also mentioned secondary sources. So many of the components of the manufacturing process have a set of foreign particulate inherent to themselves. So what I mean by that is say, for instance, plastic from a disposable storage bottle would be the primary source, but how truly clean is that vessel? The storage bottle from the vendor could add particulate from its manufacturing process into the manufacturing process it is being used in. So these are secondary potential sources for contamination that can't immediately be discounted. Something else to think about here is that there are going to be different levels of risk associated. So for example, packaging might not be important for a parenteral product, but it would be of crucial importance for a solid dosage form because that will be in direct contact. So this industrial forensic concept lends efficiency to the evaluation process in that multiple compartments can be evaluated at the same time. And again, it provides a way to make the evaluation and the data gathering more manageable while continuing to maintain the core quality by design principles. I wanted to point out here that this approach was highlighted in Pharmaceutical Manufacturing magazine, and it was published in a two-part series. The website is here if you're interested in reading more in detail. So far, industrial forensics has been a proactive approach. However, it's unique in that it can be applied retroactively to an already established process. There are two schools of thought here. So the first one, start from the end, so the packaging or fill finish operations, because this is usually where you have the most risk for contamination, and work backwards to create a list or a database of the potential sources. The positive here is that you are still using the systematic approach for evaluation, so you will keep organized. But the negative is the lag time that it could take to amass a comprehensive characterization of all of those process materials, and in turn this can lengthen investigation times initially. The second approach would be to take your finished product and work them up to identify the types of particulate within. So you would use this data then to drive the identification of source materials of the same nature as the contamination being identified. So for example, you find a clear fiber, so you go back through the process and look for other types of clear fibers as potential sources. So the positive in this case is that you are building a smaller database and you're quickly characterizing items already in the product and already causing an issue to close those investigations. But the negative would be that you lose the characterization of other source materials, and then you would be back to square one if you encountered a different type of contamination. So you can also lose that systematic approach to evaluating the process, and in turn it would increase the potential for disorganization. So the best approach would be a hybrid approach. So start with what is already in the product to quickly identify things that are causing immediate issues or are at high risk, but in parallel continue to use the industrial forensics approach and build up a database of additional materials to facilitate all investigations. One other thing to think about here is having a vehicle for searching assistance. So you're going to be collecting a large number of data points, both your list of process materials and where they came from, and all of the other characterization data. A data management system is going to be necessary, and you'll need something that allows for both sample information searching as well as the ability to query for technical data. So a versatile data management system can add to the efficiency of root cause investigations. At this point, I'd like to go ahead and share a few case studies in order to highlight the foreign particulate characterization regimen that we've discussed. These case studies will build upon one another by demonstrating the way an investigation would unfold with a retroactive versus a proactive approach. So in this first case study, there was a bottle of liquid sent for investigation. The pharmaceutical company had indicated that there were needle-like particles within that had appeared during a stability study. During that stability study, the bottle had been incubated at a very high temperature. So the first step for analysis is to extract and prepare the particles of interest. So in this case, we used a filtration isolation technique. Here in the image, you can see a glisten of the particles that are lying on the filter that was collected. They were only a couple microns in width. We then subjected these particles to the forensic characterization regimen that we've discussed. We utilized polarized light microscopy, and this provided additional information about the crystal-like structure of the needle particles. For the infrared analysis, we used a reflectance micro FTIR technique and obtained a spectrum. For those of you who are not familiar with the FTIR, the peak annotations that you see identify functional groups in the material, and then that's what helps you to make an identification. For the scanning electron microscopy, so SEM, again you can see the crystal-like morphology at very high magnification. Utilizing the energy dispersive spectrum that you see in the bottom portion of the image, there were elements of carbon and oxygen present. So we knew in this case study we were dealing with something that was organic. In this case, the FTIR spectrum was unusually complex, so in addition we chose to also analyze by Raman spectroscopy, and you can see the Raman spectrum that's displayed here. So for the characterization identification, we searched the IR and Raman spectra against databases and got a hit to a material identified as BNX MD1097. At this point in the investigation, we decided to obtain a reference or a standard of this BNX material to subject it to the same types of analysis for comparison purposes. As you can see, the red spectra in both the FTIR and Raman results is representative of the sample, so the needle-like particles, where the blue spectra were obtained from the standard reference material. It doesn't take an expert to look at those results and conclude that they were nearly identical. The next step was to further investigate this material. We did some research on the BNX material and obtained the chemical name of the compound, and we also confirmed that it's commonly used as a polymer additive. At this point, we went back to the company and we presented these results, our findings. Through the discussion, it was suspected that these particles were possibly leaching out from the bottle containing the liquid during the incubation because of that elevated temperature. The company then went to the vendor of the plastic bottles. The vendor indicated that the bottles contained an oxidizing agent used as a polymer additive that was nearly identical in chemical structure to the BNX material. So therefore, for subsequent studies, the company resolved this issue by simply utilizing a different type of plastic bottle. For the second case study, you'll see another example of the characterization identification in a retroactive approach. The issue in this case was a particle that was observed in a parenteral product. The vial itself and the corresponding syringe were submitted for investigation analysis. The company was suspecting that the particle may be consistent with the septum material. In the image, you can see the particle kind of stuck to the side of the vial here, encapsulated in the product matrix. So we extracted the particle from the product, rinsed it with water, and prepared it for the forensic analysis. An optical image was collected once we isolated the particle. The stereomicroscopic evaluation revealed a particle that was gray, and it measured approximately 700 micrometers in length. When probing the material, the observation of it being soft and rubber-like was recorded. FTIR analysis was performed and the spectrum you see was obtained. This is consistent with a butyl rubber-based material, and it contained indications of silicates being present. The SEM-EDS analysis revealed a carbon-rich matrix with elements of aluminum, silicon, oxygen, and sodium. When it was further investigated, it was determined that there were small inclusions that were rich in silicon and aluminum, indicating there was an aluminum silicate filler material, and this is something that we commonly see in rubbers. So the characterization revealed the butyl rubber with aluminum silicate as possibly consistent with the septum material. However, we wanted to be able to confirm this. The next step was to analyze the septum material, subjecting it to the same forensic characterization regimen that we did for the gray particle, the foreign particle. So as you can see in the data comparison, the particulate and the septum were chemically and elementally similar. The question to think about at this point is how to definitively identify the source. So are there additional materials in the process that are similar in composition? At this point, we took it a step further and continued investigating. It was noted that there was a missing portion of the vial septum. The company did indicate to us that the septum had been punctured with the syringe needle, so this was consistent with the piercing. We transferred the original particle and utilized another forensic technique known as fracture matching. And the particle was oriented into the void. As you can see at high magnification, the particle edges lined up with the void. So essentially the particle fit in like a puzzle piece. The combination of the forensic characterization then with the fracture matching allowed a conclusion with a high degree of certainty that the gray particle sourced from the septum. And we presented these results to the manufacturer, who immediately initiated an observation investigation for the administration technique of the product. To resolve this issue then, additional training on proper administration technique was required. So the problem was solved, but it was retroactive versus proactive. A proactive approach could have saved a lot of investigation efforts. So if it was known ahead of time the composition of the septum material, so that we would not have had to take a step back and analyze that. And secondly, if it would have been known that there were no other butyl rubber materials throughout the process, such as a gasket or a seal, in which the industrial forensic concept suggests. This third and final case study will show how a proactive approach makes for a quick investigation. So the issue in this case was a particle that a company